Fire, 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 fire. In order for a cell to maintain homeostasis, it has to regulate both the type and rate of chemical reactions that occur in the cell. It does this by using enzymes. So today we want to talk about enzymes and how they regulate chemical reactions in the cell. We'll have a few learning outcomes. The first is to explain a couple of types of chemical reactions that occur in the cell. The second, we want to investigate the role of enzymes. We're going to describe the structure of an enzyme and how structure relates to function. And finally, we want to summarize how enzymes are turned on and off, or how their activity is regulated. Let's start by talking about two different types of chemical reactions. Some chemical reactions release energy as they proceed, and some chemical reactions require the input of energy to go forward. One of the reactions we might be familiar with is eating spaghetti or starch. We know that if we eat starch, that we can use that energy to power our activities like muscle contraction or ion pumps and other things. All starch is, actually, is a bunch of glucose molecules hooked together. So a bunch of simple sugars linked together with covalent bonds. And there's energy stored there that, together with oxygen, can be released and wants to be released. So the glucose burns in the presence of oxygen to produce CO2, water, and energy. So I can start this reaction by adding heat, what we call activation energy. And once this piece of spaghetti is lit, it will continue to burn as long as there's glucose and oxygen around. At this point, it's a slow rate. I don't know if you can still see the flame, but it burns just as we'd expect. So the stored energy in glucose can be released through digestion in our body, or if we simply burn it. Now, some chemical reactions don't release energy but require energy, and those are called endergonic reactions. So I'm going to use an example of this flashlight. If I spin that light, or I spin the crank, I can produce electricity that can be converted to light. But as soon as I stop spinning the crank, the reaction stops. It's not like burning spaghetti that releases energy. It continually requires the input of energy to move forward. In our bodies, the most common source of energy is ATP. So a chemical reaction might go forward, but it might require the input of energy. And as we'll see, an enzyme will use the energy from ATP to push that reaction forward, even though it wouldn't otherwise go that direction. You may be familiar with another type of material that burns. This is a piece of wood, or cellulose. You'll see that it burns very much like spaghetti. As long as there's oxygen around and there's wood left, this will continue to burn. Now, you might say to yourself that those are two completely different materials, the starch in spaghetti or the cellulose in wood, but actually what we're going to find is that starch and cellulose are the same material. They're polymers of glucose, or just a bunch of glucose molecules hooked together. The difference is that a simple starch is formed from alpha glucose and cellulose from beta glucose. So the bonds that hold these two molecules together are different shapes, and that's shown here. It turns out the reason we can digest starch and not cellulose is because we have an enzyme that can break these covalent linkages between the glucose molecules and starch, but we don't have an enzyme that can break the bonds between the glucose molecules in wood or cellulose. Okay, let's have a look at how enzymes work. Enzymes work by lowering the activation energy it takes for a chemical reaction to go forward. All that means in words is this spaghetti doesn't have to be hundreds of degrees to burn. It will burn in my body at body temperature because an enzyme will help the reaction go forward. How does it do that? Well, remember, this is the image of starch, the diagram we saw. And you can see this bond is triangular. What an enzyme can do is it has an active site, this lock and key formation, so the inverse triangle. And the enzyme can go up. The active site will associate with atoms in the bond it wants to break. And it will make the electrons around those atoms and those bonds happy as the electrons are shuffled and some bonds are broken and others are formed. Now let's review, take a look back at the structure of cellulose. Cellulose doesn't have this triangular structure to the bond. So the enzyme amylase that can associate with the atoms and electrons in the alpha linkage between the glucose molecules in starch, that enzyme can't associate with the atoms in the beta linkage between the glucose molecules in cellulose. So this is another example of the relationship between structure and function. If the structure of an enzyme is such that it can bind to a certain bond and stabilize those electrons, then it can be an enzyme that catalyzes a reaction, and if it's not, it can't. So imagine if there were some mutation or a shape change in this amylase, and it could no longer associate the way it normally would, then it couldn't break the bond between the two glucose molecules. In this case, we couldn't break down the starch and get energy from it, couldn't eat spaghetti, and so we couldn't derive energy from that molecule, from that substance, just the same as if we ate wood. Let's have a look at ATP can help catalyze an endergonic chemical reaction. A good example for us is the sodium-potassium pump. At rest or at homeostasis, sodium levels are really high outside cells and potassium levels are high inside cells. There's also a little bit of a charge difference. But at rest, sodium would want to leak into a cell and potassium out, and that will continually happen. The only way to maintain high sodium levels outside the cell and potassium inside the cell is to use energy to pump those ions in a direction they don't want to go. So let's have a look at a pretend sodium-potassium exchanger, this little mouse trap I have here. I have three sodium ions, they're Kick cereal, 2 potassium ions that happen to be cashews. And at rest, the sodium-potassium exchanger can go ahead and bind to these sodium ions that are inside the cell. If ATP comes along, it can both cock and release those sodium ions outside the cell. Then the potassium ions from outside the cell will bind to the sodium-potassium exchanger and cause it to flip and change shape again, and then be released in the cell where the sodium ions can again bind. And if there's ATP around, that can cock and spit them out of the cell and then flip back in with 2 more potassium ions. Hopefully that made sense. The mousetrap is an imperfect metaphor because it shows the pump flipping back and forth in the membrane. The main point is ATP can force a change in shape in the protein, which can push sodium and potassium in a direction they don't want to go. Now, I said at the beginning of the video that a cell needed to regulate both the rate and type of chemical reactions in the cells. That implies that enzymes have to work at different rates at different times. We can't always have the same chemical reactions happening at the same rate. So how does that regulation happen? Well, it can happen because we regulate the amount of an enzyme that's there. Most enzymes are proteins, so if we turn on the gene that encodes that protein, we'll have more of the enzyme. And if there's more of the enzyme, the chemical reaction will go forward. An example of this includes genes regulated by a lot of steroid hormone receptors, like the aldosterone receptor. the aldosterone receptor. Aldosterone is a hormone that will increase blood pressure and blood volume. And it does this in part by increasing the amount of sodium that's pumped back into the bloodstream in the kidney. Now, when aldosterone binds to its receptor turns on the gene for a sodium-potassium exchanger that helps pump the blood back, that helps pump the sodium back into the bloodstream like we saw before. Other enzymes are always around but waiting to be activated. So think of the enzymes that promote blood clotting. If we need a blood clot to happen, those reactions need to occur very quickly, and they can occur quickly because enzymes can be activated quickly. In blood clotting, an enzyme like thrombin is present, but it's inactive. And the reason it's inactive, it's a form called prothrombin, where the active site, like the active site in these scissors, is covered up by a piece of the protein. Now, when other enzymes are activated, a protease comes along and clips thrombin so that regulatory part of the protein is no longer there. And then the active site on thrombin is free to go around and clip more proteins and activate them. So we see the theme of structure and function again. In the case of thrombin, the inactive shape is present, and the active shape becomes present only when other enzymes turn it on or help change that shape. That's called post-translational modification of a protein. The protein is there, it already exists. The gene has been transcribed, the mRNA translated, the protein is around in an inactive shape, but some signal can cause a change in shape to activate the enzyme. Post-translational modification. Okay, let's see if we can summarize what we've been talking about here today. First, chemical reactions may release energy or require energy to proceed. Enzymes control the rate and type of chemical reactions that happen in a cell. So even a chemical reaction like burning glucose or burning starch won't happen on its own without the input of energy. But whereas we need a lot of heat energy at room temperature to release the energy stored in starch, in our bodies, that energy can be released very quickly because we have the appropriate enzymes. But the enzymes have to have the correct shape to catalyze the chemical reaction. So even though starch and cellulose would have the same chemical structure in terms of the glucose molecules, because the bonds have different shapes, the enzyme that breaks down starch will not break down cellulose. Therefore, we can digest starch, but not cellulose. So the structure of an enzyme is critically important to its function. And then finally, enzyme activity can be regulated either by regulating the amount of the enzyme that's present or the shape of the enzyme, which in turn regulates its activity. The amount of enzyme present in a cell is regulated at the level of the gene. Many enzymes are proteins, so if we turn on the gene, we'll have more of that enzyme. On the other hand, some enzymes are always present, but they can be turned on or off by changing their shape. So hopefully you got some good information from that video, and we now understand how cells and tissues change their function by changing the rate and types of chemical reactions that occur within the cells.